Cool. Hello, everyone. Welcome to April's APAC Scientific Forum today. So for today, we do have a pretty packed agenda. So if you can go to presenter. Yeah, thank you. So first, we want to introduce our STAR team from Singapore. And then next, we'll really go through kind of our main topic today. So we'll have a study package review by Chen and Hong Kong UT, followed by the AMIA abstract review from Changsu and Sujin. And last but not least, we'll continue as the SOS challenge has kicked off last week. So I'll just want to also give everyone an update what has happened so far, and also, of course, call for your participation. So with that, we are very glad to introduce a STAR team from Singapore. So we have Mukesh here, who will be helping with all the APAC Scientific Forum this year. So Mukesh, if you could, let's say hi to everyone really quick. Welcome.
Thanks for the warm welcome, Jing. Hi everyone, I'm Mukesh from the A-STAR team here. My team looks after the data and analytics component of the research we do in A-STAR. So I have also Cindy and Macy as well. Maybe they can also say hi to the team.
Hi.
Hi, I'm Cindy. Can you see? Hello. Yeah, so I'm part of Mukesh's team. Yeah, doing some mapping for our study, for mapping to OMOD for our study. Yeah.
Thank you. Nice to meet you.
Hi, I'm Maisie, also from Mukesh's team.
Hello, nice to meet you. Cool. Thank you. Thank you, Mukesh. Thank you, Cindy, and thank you, Messi. So next, we really look forward to working closely together in the next, in the future with you. So next, we will really go into our main agenda for the day. So first, we'll have a study package review by Chen. So Chen, I don't know if you want to share your screen.
Yeah, let me do that. Please.
All right,
wait
thank
a little
you.
bit. So, yeah, this is brief update of chapter, the characterization of health biology APAC chapter to identify temporary effect of the pandemic. And the main role of this package is to calculate the incidence, and those are the method to calculate incidence or prevalence. It is much easier to calculate prevalence compared with the calculating incidence rate, because to calculate incidence rate, we need to denominator of time each person was observed total for all persons. That is quite different between the incidence rate and the prevalence. And since I'd like to calculate the monthly incidence, it was not easy. So let's assume those are the observed population in our databases. So I'd like to calculate the incidence of some diseases between January 1st, 2021 and January 31, 2021. So among between these during this 1 month, about 31 days, I'd like to calculate the incidence. Then I need to calculate the time at risk of the population across this time span. So I described 5 patients. So first one observed from the 2020, and then the patient had the outcome at this point during the January of 2021. The second one started observed after 2020 January, and then observed later after 2021 January. The third one has more. Third one had lesser observation period, and fourth one, who started to be observed from the 2020, and the patients got the outcome during this month. And then last one, who did not have the outcome, and then the observation period stopped during the January 2021. Among these patients, I want to filtered those who observed more than 1 year before my time spent at interest, and then observed, but those who observed, those whose observation period stopped earlier than the 31st January actually had survived in this denominator cohorts, and that's what I've implemented in my package currently, and let's show you how I did it. Usually, when I develop a package, I fork the package to my own GitHub repo. So my main repo is located in the Odyssey studies. The name is chapter. And then I usually, as I said, I usually fork this GitHub repo to mine. In this case, I fork this package to Dr. Yu group. So I worked in this package, in this repo of Dr. Yu chapter repo. And you can see how I did it, add these functions in the Git commit history, and then these are the Git commit how I did. I added the base population to create the denominator cards. So in here you can see that the SQL code are located in the inst folder. So I added this create course SQL file in here, and you can add some functionality to make them if-else functions in the SQL codes by using this theme. So in the bracket, you can, if it is true, then the following line will be executed, and if it is false, then the second line will be executed. Those, it's how we can implement if-else function in the SQL script by using R. And this is the R code for this to execute the SQL code. So now here I made this. I packed all of these functions into a package, and then you can, if you activate this function, then you can make the denominator codes, and then so now I can calculate the time and risk of this denominator during the month, and then I will make the outcome codes to calculate incidence rate. So this is the progress of mine so far. If you have any comments or questions, please let me know.
Thank you, Chen. Any questions from the audience?
I know it's quite new, and since I need to add novel functions to the current Odyssey tools ecosystem, it was not easy, and it takes too long time for me to make a package. But I think that you can learn how to develop your one function in the Odyssey tools ecosystem by reviewing my comments. So please review my comments, and if you have any questions, let me know. If you can write your question in the issues of GitHub, or you can send a mail to me. Okay, that's it. That's my current progress.
All right, thank you, Chen. So any other questions that the audience, the team may have? All right, so a thumbs up on morning. Thank you. Thank you, Chen. All right, so next we'll have Hong Kong U team to can introduce and have everyone to go through together the study package. So I see Chai is here. So Chai, or Ivan, I'm not sure like who I'll be presenting first. If you could.
Yes, hi Jing. Yeah,
Hi,
this is Ivan.
hello.
I'll be presenting. Just one moment.
Awesome. Okay.
Hi, Chan. Song here while Ivan is preparing for his preparation. So, for his presentation. I'm recalling like some of the conversations we had in or previous study calls. I think you mentioned that the incidence needs to be changed to calculate on a monthly basis. Is that reflected in this new update that you made to your study package?
Yes. So currently, you can calculate monthly or yearly, or just
Okay.
you can calculate incidence based on your your definition of the time span.
Okay. Great. Thanks.
Thanks.
Sorry, Ivan.
Right.
Sorry I took your thunder. Take the
No
floor.
worries. Yeah. No worries. Yeah. So good. Good afternoon. Good morning to everyone. So today, I just want to give a review of our of the progress of developing the study package for our study, which is the study number 2, which is coded as a KISO. So first of all, before we go into the study package, I just want to give you guys a reminder of our study design. So for this study, we aim to investigate the short, medium, and long-term outcomes between patients with a COVID-19 infection, with COVID-19 infections, and to those without. So this will be a retrospective cohort study. So the first step in building the package would be to define to come up with the definitions for the COVID-19 infection. So in the end, we have settled on the current this definition that I'm showing. So these for patient with a COVID-19, we would define them as those who have a positive test results or a COVID-19 diagnosis after the 1st of December 2019, and with a continuous observation of at least 365 days prior to their COVID infection. And they should not have negative test results within 3 days after their positive test result. And this is this definition has actually been adopted from another Odyssey study on COVID. So yeah, just to keep and make sure our definition is consistent with other Odyssey study. And so this is when we use Atlas to define cohort for this study. And this is how it looks like when we put in the criteria that I've mentioned into Atlas. And for the comparator cohort, this consisted of patient without a positive SARS-CoV-2 test result, and also who do not have a positive test result or diagnosis for COVID-19. And again, we use Atlas to define these cohorts. And this is just an illustration of how it looked like when we put in these definitions. And as you can see, we set them, we define those who this group of patients as those with exactly zero occurrence of a positive test result from their COVID-19 tests, as well as exactly zero occurrence of COVID-19 as a condition. So yeah, after building the two study cohorts, we can move on to doing the propensity score matching step, which aims to minimize the confounding bias in our study results due to any differences in the baseline demographics between the two the two cohort for comparison. And this step was actually quite tricky when we tried to assign an index date to our comparator due to the lack of a distinct event which we can anchor our the start of the observation period for this group of patient. So in the end, we have came up with this method which enabled us to assign a pseudo index date to our comparator as their match control as their match target patient. So in the first step, we would match comparator subjects to subjects from the target cohort based on their year and month of birth, as well as their gender. We excluded those who whose index date is outside of the observation window of their comparator. So we excluded those whose index date was after the observation period end, or if their index date is 365 days before the observation period. And this is to ensure that the pseudo index date assigned to the comparator would be within the observation window of their target, as their respective target patient, and to ensure that each comparator would have at least 365 days of observation period before their pseudo index date. And after excluding this group of comparator, each comparator remaining would be mapped to a target patient with the closest observation start date. And if there's more than one comparator which fits all of these criteria, then we would randomly select the comparator for each target patient. So after assigning the pseudo index date for each for our comparator, we can move on to estimating the propensity score before doing the matching. So this is how we conducted the propensity score estimation, and these are fairly we use the standardized method that is available in the Cyclops package from which is an existing Odyssey package. So this this package uses the large-scale regularized regression model based on a long list of generic characteristics, including the diagnosis, drug exposures, measurement, or any medical procedures that has been undertaken in a patient before the day of exposure, and this estimates the propensity scores. And afterwards, we would conduct the matching. We would match patient from the target to the comparator with a similar propensity score. So we use 1 to 10 matching with a caliper width of 0.2. And again, after we've put in.
Sorry, may I ask? Hi, morning here. Yeah,
Oh,
so for the matching, may I ask a quick question about the matching?
Yeah, sure.
So I guess 1 to 10 matching because we have very imbalanced data. Is it correct?
1 to 10 is we try to maximize the not imbalance, but we try to maximize the study population. So we want to maximize the number of patient from the two cohorts. So it's actually 1 to up to 10 matching.
Also for both case and control, they can match up to 10.
No, so it's 1 from the target cohort and up to 10 controls for each target.
Okay, so okay, I guess you want to maximize the usage of the target population, I guess.
Yeah, and we want to maximize the number of comparator as well. So each target can be matched to up to 10 controls.
Okay, and the caliper set at 0.2 is on the high side, right? 0.2 is quite a high caliper.
I think it's also. I would have to check on that, but when we define these criteria, these were, I believe, the standard settings that was used in the within Odyssey. So yeah, but I will have to double check on that. But it seems yeah, I believe it's yeah.
Okay, sure, no problem. Just curious,
Yeah, I can double check on that as
Okay,
well.
thanks.
Ivan, it is standard. 0.2 is by default. So if you, in case you need confirmation on that, yes, it is standard.
All right. Okay. Yeah. Thank you, Song.
I mean,
yeah.
Song, thanks for asking. I mean, can we trace the history, like why this was set to be the default?
I think some more scientific people can answer this, right? Like maybe Chan can help out. I don't know. Sorry to call you out, Chan.
Just my personal humble experience, 0.2 is quite high. Usually on the higher end, yeah. But anyway, yeah, the others may disagree. Yes.
I think the default.
Good thing to put in. Good question to put in the forum and ask.
Yeah, the default width caliper width will be 0.2 as long as I remember, right? The default setting is 0.2, right?
Yep, correct.
Yeah,
I think that was used in the legend work as well, I think, Chan. So 0.2, I think, is standard.
Yeah. So previously, I tried with the narrower width 0.1, and it did not make a better outcome. And at the time, I searched the papers about the which width of caliper would be best, and to my knowledge, 0.2 is kind of the standard. You don't know, you know that there is no real empirical evidence for the caliper width, but 0.2 was the kind of the default of the paper, many papers. So yeah, it's not that big in my experience.
Cool. Thanks, guys.
Yeah. So perhaps I'll move on from here. But if there's any further suggestions, or if there's any other evidence which suggests other values for the caliper, or if 0.2 is in fact the go-to values, then please do let me know. Yeah, and so this is like this is how it looks like when we inputted these settings into Atlas as well. And on top of the two comparative cohort we created, we also incorporated more than 90 disease outcomes under these involving these organ systems as outcome cohorts into our study package. And for the full list of the outcomes for this study, please do refer to our study protocol, which is available on Teams. And here is an example of how of the concepts we use to define each outcomes. And these definitions have been adopted from either from other Odyssey studies or from the Phenotype February campaign from the community. And so, yeah, on our study package also included two subgroup analysis based on the age group and gender of the study populations, and we included a list of negative control outcomes, which were believed to have no causal effect from COVID-19 infection, and that was and these were used for empirical calibrations of the hazard ratios and the confidence intervals of our results. And yeah, and here are some of the examples of the results. So from the table above, you can see that we estimated the hazard ratios and the calibrated hazard ratios for each outcomes. And here is one of the outcomes as an example. So yeah, here's the hazard ratios and their respective confidence intervals. And the table below shows the number of subjects that were included for these comparisons, their follow-up period, and the number of events, as well as their incidence rate for this particular outcome.
Ivan, how did you define the negative control outcomes here?
Sorry.
So to calculate the calibrated hazard ratios on confidence interval or p-values, you have to use negative control, which is not related with the COVID-19.
Right.
And
yeah.
Since we do not know which diseases or conditions are not related with COVID-19, so it was not easy to define the negative control outcomes here. So how did you define the negative control outcomes in this study?
That's a good question, and it's actually quite tricky when we try to come up with the list of negative control as well. So the quick answer would be we looked at other Odyssey studies on COVID-19 as well, and we tried to select those who that is sort of obvious that should not be affected by COVID-19 infections. But again, we cannot be very certain that these were in fact negative, have no association with COVID-19 infection. So at the moment we are interpreting, we're still deciding whether in fact the non-adjusted hazard ratio would be more suitable to evaluate the risk following COVID-19. So yeah, it's still sort of open for discussion whether we can be exactly sure on that. Yeah, so I hope that answers your question.
Yeah, I'm gonna have a quick question. I think it's more on the data source. Yeah, perhaps you might have covered it in the earlier forum calls. What's the difference between the IQVIA Italy original data source and the updated data source? You can see that it changes your hazard
Okay.
ratio.
So yeah, actually, please ignore the updated. The data source is the same. It's just we tried we tried another method that's which is labeled updated here, but we decided that there was some issue with the newer method, so we have reverted back to the original design for this study, and that's why I've highlighted the original tab up here. And so it's the data source is the same. It's just we trialed another study design, and we've decided the original study design would be best. So hence, yeah, we've chosen to present the results here. Yeah.
Oh, thanks for clarifying.
Okay. And here is the flow chart to show how we selected the study population for each disease outcome. So yeah, as you can see, we excluded those with a prior outcome of that specific outcome of interest. We excluded those with more than 1 day at risk and those who cannot be matched based on the propensity score. And finally, here is just a graph, a figure showing that the covariate between the two study cohort were actually fairly well balanced between the two target and comparator cohort after the propensity score matching. So I guess this also supports that the caliper width issue that perhaps 0.2 would be suitable as we can see that the covariates are fairly well balanced after the matching. So yeah, but I guess it does support that the caliper width used to when we match the targets and comparator by propensity scores is sufficient to achieve a well-balanced study population.
Ivan, are you still here with us?
I cannot hear from him either. So
Oh, sorry.
Oh yeah,
Can
you're back. Yep.
Oh, sorry. Should I go back a few slides? Yeah, so here is a flow chart of how we derived our study population for this study. We excluded those with prior outcomes, with those with less than 1 day at risk, for the particular outcome of interest, as well as those who cannot be matched on by propensity score. And here is a figure just showing that after propensity score matching, the covariates are fairly well balanced between the two study cohorts. So I guess this also supports that the caliper width of 0.2 we use in our study design is sufficient to achieve a well balance of the between the two between the targets and comparator cohort. And finally, I just want to acknowledge the help from Xiao Yu and Chan from IQVIA, as well as my colleagues for their help in putting this package together, and their support. So that would be it for my presentation today. So if there's any questions or suggestions, then feel free to let me know.
Thank you, Ivan. I think we have a hands raised. Atif?
Yeah. Thanks, Ivan. The 2 quick points. I think the first one is it'll be important for you to kind of maybe exclude patients who are taking Paxlovid or monotherapic antibodies, because I know personally that you can get negative after 3 days if you start one of those therapies early enough. So it naturally comes off as a patient might be excluded in your study. The second point is, I think there's a correlation you're trying to get with here, which is patients who've been infected with COVID have some sort of inflammatory or pathological response to be higher risk. But there's also the pandemic case where changes in the health system, visitations, etc., has also led to variations in risk. For example, in diabetes, and this is well established, that there's an inflammatory cause of COVID making you having higher glycemic risk, but also patients, because of lack of glycemic care, telehealth, there's an increased incidence of patients having hypoglycemic attacks as well. So I think there's a limitation section you probably want to put in that, because we haven't done either adjusted for that at the propensity stage, or we haven't done the mediation analysis. It's hard to distinguish effects from the infection itself versus effects just because of the pandemic disruptions in the health system. It could be either or. And for certain diseases, because you're looking at 90 different outcomes, some of those impacts might be a bit more on either end of the spectrum.
Yeah, I think, yeah, thank you for your suggestions. I think the impact of changes in healthcare policy would certainly be a limitation of this study. But in terms of the clinical, the clinical cause for these, for any difference in terms of the risk of clinical outcomes, I guess our propensity score matching step does to an extent adjusted for not just accounted for such difference, and we try to make sure each the target and the comparative cohort are well balanced in terms of their the baseline demographics of patient is balanced before we conduct the comparison. So I'm not sure if that answers your.
It does, yes.
Okay. So thank you. If there's no further questions, perhaps I can end my sharing here.
All right, cool. Thank you. Thank you, Ivan. Cool. And next we'll have Chongsu and Sujin. I think Sujin is here today to give us a quick update on the AMIA abstract submission that we just did, I think, by end of last month. So Sujin, if you can share from your side. Sujin, you're on mute if you're talking. You're on mute. No problem. Yes, we can see the screen.
Yeah. Oh, Hi everyone, I'm Susan studying at Aja University and I'm here to present on behalf of Chong-soo as he unfortunately couldn't make it to this meeting due to other commitments. So today I will tell you about our submission of CDM inspection research to the AMIA conference and our plans for future submission very briefly. So on March 21st, Chong-soo and I submitted a research poster on CDM inspection to the 2023 AMIA conference, and we are so grateful for your help along the way. And thanks to the support of the Wonders organization for this AMIA submission, we were able to update our research to include the analysis from Wonders that were missing in the previous research. So this is our plan for submission of the CDM inspection research. Chong-soo and I plan to finish this CDM inspection paper manuscript by July, and we will keep you updated on our progress, new findings, or developments as they arise. So by our next meeting, we will have the methods and research sections of the paper written and shared with you. And we greatly appreciate any comments or suggestions you have on our work at that time. Thank you again for your support and helping us to successfully submit the poster to AMIA. So here's my presentation. So if you have any questions, please let me know.
Hi, Sujin Song here. Do you know when the acceptance will be circulated to everyone who submitted?
I think it was May. I will check it out and share with you guys through
Okay.
chat. Yeah.
Sounds good.
Thank you.
Thank you.
Looking forward to hear from that.
Okay.
Thank you,
Atif,
Sydney.
Atif, is your hand up from the last question you had, or is it a new hand up?
It is, sorry, yes. Nope. It's the same hand from last time. I put it down.
Okay.
Hey, morning here. Yeah. First of all, yeah, thanks for putting up the abstract and for the submission, and glad to hear that we are targeting a manuscript submission in July. Just want to clarify the team. If any institute would still like to join the study, is it too late now, or I don't know, or is there a deadline when the new institute may join or they may no longer join?
It's not determined yet, and maybe I can ask Chong-soo about it. And if you let me just, if you list up any other organizations you want to join, then I will ask him.
Sure. Because I was initially thinking that to use the AMIA abstract as a promotion material to circulate to the other institutes in Singapore, trying to convince them to join. But I hear you now that you plan to finish your manuscript by July. So I'm not sure. Yeah, please let us know that internally. When is the deadline that we stop taking in anyone, that I will let them know. Maybe it's good to give them some urgency as well, saying that, if you guys are up for it, please respond and finish your deadline by this deadline, then otherwise we will move on without you.
Okay, but we haven't started writing yet, so I think, I don't know, but I will ask Chong-soo.
Sure. Yeah, just provide me the deadline, and also, if I may, also you can confirm with us that whether we can use the abstract because it hasn't been published. So can we use the current draft of the abstract, as I mentioned, as promotion material and circulate with our partnering institutes and try to convince them to join?
Oh, sure.
Okay, thanks a lot.
Thank you. So any other questions? Okay, thank you.
All right, thank you. That's all. Thank you, Sujin. So that will close all of our study updates from the owners today. So we have another item, not item that we want to discuss with everyone today is around SOS call. Sorry, not SOS call, SOS challenge. So with that, we want to just quickly go through that with everyone again. Let me share my screen really quick. So right, as you may have been hearing about it for the last 2 or 3 weeks. So we just want to quickly share again that SOS challenge has kicked off as of last week. And then we're on week 2 for this week, which we did go through the data diagnostic. Can you see my screen?
Okay.
And I will also, okay,
Yeah,
cool.
See you.
And I will also, okay, so cool. So we want to mention, so last month we kicked it off, and then this week we're discussing the data diagnostics for both the US time and then APAC time. So for us, it would be 8 a.m. on 8 a.m. Korea time on every Wednesday, starting from last and today. Yesterday was the second. We'll kind of keep continue going till another 7 weeks. So that's on the logistics of the call. And of course, kind of there are always office hours and also the recordings from the previous from the previous meetings that you can kind of go through and let us know if any questions you may have. Oh, did I raise my own hand?
So we recommend data partners to execute the DB diagnostics. We'll evaluate the databases and then decided which databases will be feasible for 4 SOS challenges. And in this week, we introduced how you can run and execute DB diagnostics, which is led by Jing in this week, and you can see the recordings in in the YouTube too. So please have an interest in this SOS challenges. And
does everybody here? Sorry, does everybody here even know what the SOS challenge is? I don't know if people do, because not every. I know most of these folks aren't even on the SOS challenge calls. So to back up, SOS challenge is actually effort done by the Global Odyssey, right? So Morning, you talked just about wanting to get your other Singaporean organizations to be part of something. This is an opportunity that I think we should push all of our collaborators to be part of, right? The SOS challenge is basically something that happened in February where people were asked to submit questions and the community voted on it. I know you guys got an email to say, hey, vote for the one that's in APAC, but there's actually 4 of them that was picked, and all 4 of them, if you go to this page, it's at the bottom here. These are the 4 that we're talking about. Okay, you got this one that's on VEGF. We got the fluoroquinolone. I still can't say this, even though I did this yesterday. Okay, and this one is the one that's the APAC one, right? This one over here, you can see it's the APAC one. But then we also have another one on. This is, I think, ischemic stroke. And then this one is on PML. I could do PML and MS very easily. Okay, so they're all 4. And what the SOS challenge does is basically we're teaching people in Odyssey how to actually conduct a study, conduct a network study. And we're doing that through via a series of tutorials. Okay, the first one, and they happen on 2 different times, right? And they did this so that they can catch the APAC side as well, right? So you have our normal community call at 11 a.m. Eastern Standard Time, the global one. And then now there's a 7 p.m. Eastern Standard Time, which is 7 a.m. Singapore time, 8 p.m. Korean time, 7 p.m. a.m. China time, right? That is really more APAC friendly. And those are going to be taught by APAC folks if we can get them. If not, they're taught by global folks, right? So last week, Chan went over how you even set up a network study, okay? And if you want to watch that video, it's down here, okay? You can watch 2 versions. You can watch the version that's taught right in the morning by the global team, or you can watch the one that Chan taught, right? And this week, what we went through is data diagnostics. So now you set up the study. What's the next thing? We want to know which data asset best fits our analysis, right? So that's what we went through yesterday. And if you want to do that yourself, and this is where Chan's saying call for participation, right? We want all of you guys to run these. Those that have data, we want you to run this. This is the data diagnostic package that you can run. Again, go watch the tutorial and I'll show you exactly what we're talking about. What it's going to end up resulting in is your data actually will be added to what you see here, okay? It becomes an R Shiny app. Now, caveat, data doesn't go anywhere. It's aggregate results, actually runs more aggregated counts from your Achilles heels results, okay? And you send it to Claire and team, and it'll be staged inside Odyssey Coordinating Center. It won't leave. It's only being used for these 4 studies, and it's high-level statistics. But no one will see those high-level statistics, okay? What you will see is the result of your data inside each of the study questions that are being asked. Okay, so you can see here there are actually 4 studies all already built into here. Okay, the first one that we had in the morning was this VEGF one, right? And there's 3 of those cohorts that came in. So this is the other one, and the third one is right here, somewhere right there. So now you can see very quickly, all the zeros means those are the most suitable. Those are the potentially most suitable data assets for this particular study because they didn't have any warnings. Some of these have some warnings. Not to say they're not suitable, but it's just a warning that maybe you might want to look at those, or maybe you want to change your parameter, right? So of course we're on the APAC side. So I'm going to focus more on the APAC ones, right? The fluoroquinolone, I can't say that word. I try very hard. I call medicine my 20th language, so I give up after 3. Okay, so we had 6 cohorts because we were comparing fluoroquinolone to a different ingredients, but for the indication of a UTI against the risk of erotic aneurysm. But then we also compared it instead of UTI to pneumonia, right? So there were 6. That's why there's 6 different cohorts, because it's the comparison of 3 different drugs against 2 different indications. So 2 times 3, you got 6. Right. And at a glance, very quickly, you can see, wow, all these data assets are a potential fit for this particular study. Some of these had some issues. Okay. We can actually drill down and figure out, utilizing this summary statistic report, why they fail, why they may not be suitable, right? So if I look at one of the fluoroquinolone ones and I come down here and I look at Premier, there are parameters that you can feed into this thing, right? So these are the parameters that were fed in, that we were looking for people between 2000, the study period is 2010 to 2019. We're looking for people age 35 and older. We're looking for all race, all ethnicity, all gender. We want the continuous observation period to be 365. Now, this is where it didn't pass. Observation period 365, okay? Now, reason Premier is a collection of hospital records, and in hospital records, people don't stay in the hospital for 365 days, right? So you won't get a lot of your average continuous enrollment period isn't going to be high, okay? So, but it shows that we have procedures in this particular database. We have this drug in this particular database. We have the conditions and drugs, and we have minimum max number of patients, right? We have more patients than we need to do. We need 1,000 patients at a minimum. People ask why the 1,000. This morning you're going to ask me why the 1,000, because it's actually to do a risk prediction and a propensity score. You have to have enough patients, and 1,000 is where scientifically it's enough. Anything below that, it can be, but it may not be so scientifically sound. Okay, so I went through this very fast and very quick, but again, you guys can watch the tutorials from yesterday. But what we want you guys to do, there's 2 calls for collaboration, right? One is actually join these calls and start listening in, right? Some of you guys are volunteers to coordinate some of this or to participate as part of APAC and bring this information back, right? Best time is to show up for that APAC call. Okay, and learn from there and bring back that experience to the community call, help reach out to participants, right? Work with some, work with whomever to say, hey, who wants to participate? Here's where we are, okay? I can't show it, but if you guys are in Odyssey teams right now, there's actually channels under Odyssey that all 4 of these are in the channels. That's where everything is happening. People are chatting there. There are protocols that are out there that you can review. So please go out there, review, please go out there, comment, right? Just because you don't have data doesn't mean you can't participate, okay? You can help us organize. You can help these guys with other stuff as well. Even just looking at the protocol. I'm not a scientist, I'm not a medical person, but I could look at it to see whether it might not make sense. From a non-medical person looking at it, does this even read right? And that's helpful, okay? So second ask, if you have data, you know somebody has data, we would love you to participate in all 4 of these studies, okay? And morning, you know, all 4 of these studies, when it comes from an SOS, from the community, global community call, you know these will end up with some kind of high impact journal, okay? Because you got a lot of people in here that's helping to put these guys together. Okay, so if that's not good enough incentive for you to join an Odyssey study, then I'm not too sure I can convince you anymore. Okay, so that's our call for participation. That's what we would like for you guys to do, and to go out and get people excited and get people, you know, in. I actually am extremely excited about this data diagnostic tool, because now I can actually have something I can quickly get, right, across everybody. I don't have to ask people to send me results in Atlas, okay? I can quickly get this, right? And right now you can see we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, but this was just so that we can demo it. This isn't so that we're not saying it's these 15. We want more, okay? And we're hungry for more. And I'm going to say it one more time. We want Asia Pacific to participate, and we want you guys to be part of this if you got data. If not, help Jack, help Nicole, and help Chan with this study. Okay? However you guys think you can help, we will take. There. I think I made a pretty good pitch, a sales pitch, hopefully. So, okay, everybody go out there and tell everybody that they need to participate and help us out. And we will continue to provide updates in this forum as well, right? Just in case you miss it. But again, if you miss it, go to the site and listen to it yourself. Okay, it's very easy to get to. You go to odyssey.com under events. It's the first one, SOS challenge. So morning, there is a time boxing of this one. Okay, totally time boxed it until the 23rd. Got it? Again, they don't have to have data. They can go help do the protocol review. They can help write the manuscript, okay? They don't have to have data. So even if their data is not ready, they can help with some of the other stuff. I'm sure Jack and Chan would love somebody help them write out the protocol, write out the manuscript, do the result interpretation, okay? They would love that help. Right, Chan? Right, Nicole? Yeah, thumbs up.
Of course.
Yes.
Cool.
All right,
complete so, man. Complete so, mate. Yeah, no problem. Actually, yeah, I was just discussing this with Yizhi, and Yizhi, I kind of make her agree that yeah, she will help to circulate the information to the Singapore community. Of course, Mukesh in the call as well, so we all have, yeah, no worries.
Cool. All right, that's all I had. Yes, you raised your hand. Can we help you? And then she put it down.
Sorry, I clicked it by mistake.
Shy.
All right, cool. I think we are right on time. Thank you everyone for joining us today. And as usual, we'll be posting this on Odyssey website for APAC page. Thank you everyone. Have a good day.
Thanks, guys.
Bye.